The human sperm carries the immense potential to spark the beginning of life in spite of being microscopic in size. Its intricate design and function reminds us that even the smallest cells play the grandest roles in human existence. The term spermatozoan was coined by de Graaf, and the human sperm was discovered by Antony van Leeuwenhoek. Human sperm is an elongated structure approximately 0.06 millimeters in length. It consists of 4 distinct regions: the head, neck, middle piece, and tail. The head of the sperm is flat and disc-shaped. It contains a compact haploid nucleus with very little peripheral cytoplasm. On top of the nucleus is a cap-like structure known as the acrosome, which is formed by the modification of the Golgi complex. The acrosome contains enzymes collectively called sperm lysins, including hyaluronidase, corona-penetrating enzyme, and acroin, which help the sperm penetrate the egg. The neck of the sperm is the smallest region. It contains 2 centrioles, a proximal centriole and a distal centriole. The proximal centriole enters the egg during fertilization and initiates the first cleavage of the zygote, while the distal centriole gives rise to the axial filament that extends into the tail. The middle piece of the sperm contains numerous mitochondria, which are spirally coiled around the central axoneme, the central structure running through the tail and middle piece. This spiral arrangement of mitochondria is called the nebenchron. These mitochondria generate the energy required for tail movement. A thin layer of cytoplasm surrounds the middle piece, forming a sheath known as the manchette. The tail is the longest part of the sperm. It is fibrous and contains a central axial filament with a 9+2 arrangement of microtubules surrounded by cytoplasm. The posterior tip of the axial filament is naked, meaning it lacks cytoplasmic covering. Spermatozoa do not attain full motility until they reach the vagina. They also acquire the ability to fertilize the ovum only after undergoing certain changes in the female reproductive tract, a process known as capacitation. After ejaculation, sperm can survive in the female reproductive system for up to 5 days. A human male typically ejaculates about 200 to 300 million sperms during coitus. For normal male fertility, at least 60% of the sperm should exhibit normal morphology, and at least 40% should show vigorous motility. Thanks for watching. Hit the like button and subscribe to the channel for more educational content.